So again, I want to say welcome back to the 2022 Virtual Viscosity Summit. Welcome to those joining us, and welcome back to those who just enjoyed our lunch break. Now we are starting our afternoon agenda, and we'll be starting with our presentation from the University of California, Santa Barbara, and then at 2 p.m. we will conclude with our final presentation of the day from rheosense. Before we get started, just a reminder, we encourage you to follow us on LinkedIn, Twitter, and Facebook. It's a great way to stay up to date with viscosity and rheology news and updates. And we are also happy to connect you with our staff or our presenters if you would like to connect with any of the presenters from today. Now, as we go through the last two presentations, another reminder to ask questions along the way. You can drop them in the chat pane or in that questions panel that you have, and we will answer questions at the end of the presentation. All of our presenters have set aside a few minutes at the end of their presentation so that we can answer everyone's questions, and so you can just drop them as they come to you, or you can hold off till the end, but we will answer them all at the end. And now it is my pleasure to get started with our first presentation of the afternoon, coming from the University of California, Santa Barbara, with Dr. Deborah Fygenson, as well as Nate Conrad. Now, Dr. Fygenson received her Ph.D. in physics from Princeton University. She's currently a professor of physics and biomolecular science and engineering at UC Santa Barbara. She's a leading researcher in the use of DNA as building blocks to create structures at the molecular level. Nate Conrad is a graduate research assistant for the Department of Physics at UC Santa Barbara, where his research is focused on dynamics, mechanics, and structure of DNA nanostar networks. Today, they will be presenting to us DNA nanostar networks, a model system for understanding the ultrastructural origins of viscoelasticity in hydrogels. Without further ado, it is my pleasure to pass it over to Dr. Fygenson.
Thank you very much. Not really clear. Oh, there we go. Show screen 2. And play. So, can you see my screen?
Yes.
Perfect. So I'm Deborah Fygenson. Thank you for the introduction. And I'm here with Nate Conrad, who is the graduate student who is behind all the work I'm going to talk to you about today. It's great to be here. It's been interesting to listen in on the other seminars and to have some association with rheosense and learning more about viscosity myself. So today I'm hoping to convince you that one of the DNA structured materials that we're studying could be an excellent model system for understanding where viscosity comes from. Viscosity is to momentum as diffusivity is to mass transport, or as electrical conductivity or thermal conductivity is to the transport of charge or heat. And that's not a fundamental law. It's more of a constitutive relation, but it's super useful, as you guys have all been demonstrating in the different applications that you're studying. And it made me wonder, what if we understood the detailed origins of viscosity the way that we understand them for, say, electrical conductivity? Then maybe just like a material's electrical properties reflect its composition and structure, its viscous properties, we know, tell us something about its compositions and structure, but we can predict materials' electrical properties, and we can't do that for viscosity. As far as I know, there's no generally accepted theory for the viscosity of a liquid. And I've done some literature searching, and as I've learned in this field, and I've seen that there are very good empirical models. So there's kind of plenty of predictive power. There are simulations coupled with machine learning, but all these different tools require empirical measurements to provide their parameters. that and don't really let you from a new molecule or fluid predict what the viscosity will be. So that seems to me like a great intellectual challenge. And I want to introduce you to a model liquid of a coacervate type that maybe will be useful in meeting this challenge, because this liquid, we have molecular structure and intermolecular interactions that can be rationally designed. So first, I don't know if it's necessary for this crowd, but a coacervate is a dense fluid that comes when large molecules interact with each other and separate out from another liquid phase. You see that first when they separate, they form tiny droplets that scatter a lot of light, and then they tend to be heavier than the solution that they're in. And so the coacervate accumulates at the bottom. And the liquid that, well, we are studying is inspired out of biology. So about 10 years ago, it was discovered that there are coacervates inside of cells. They're these membraneless organelles. There should be a space there. And the membraneless organelles are coacervates that are under genetic control, and they have the function of binding to other molecules in the side of the cell, and thereby either concentrating them or shielding them, sequestering them, so to give some control over the reactions that those molecules participate in. The data here from the Brangwyn lab is some of the most beautiful data on these, and was really what got started my interest in coacervates. But the coacervates I'm going to tell you about are made only of DNA, and that makes them, well, although DNA is really a biological molecule, they're very much not biological because they are completely engineered. So this is an example of the simplest building block for our DNA-based liquid. We call it a nanostar, and it has, in this case, 3 arms. It's made up of 3 different synthetic strands of DNA whose sequence, represented by the little black lines here, is designed to force a kind of interaction between the 3 strands, so that this blue strand has to interact uniquely with 1/2 on one of its parts with 1/2 of the red strand, and then the other part, the 1/2 of the yellow strand, and then the remaining halves of the red and yellow strand interact with each other to form the star shape. Because we can buy the DNA with any sequence we want, we can make sure that this is the only way in which these strands will come together. Now, in the center here, they don't quite connect. We introduce a couple of unpaired bases that are important for giving the overall DNA structure a lot of flexibility. If the molecular structure is very rigid, we found that you don't get a liquid-like state when it binds to itself. You get something that's much more hydrogel-like. And so we keep 2 A bases for each strand in the center here that we call the junction. To make these, we use temperature. We buy the DNA synthetically, and we raise it up to a high temperature so that all of the Watson-Crick base pairing falls apart. And then upon cooling, nice and slowly, the most favored state forms first, which is these individual nanostars. And cooling even more, the nanostars start to interact through regions here at the end of each arm that are sticky for one another, also by sequence design. So we call this end, for this particular sequence, the alpha sticky end, and we have many different kinds of sticky ends, which by changing their sequence can be made stronger or weaker. One important note, though, is that we make only one kind of sticky end in the nanostars that I'm going to talk to you about today. That makes the overall fluid a lot more homogeneous. So any arm can bind to any other arm. If I suppose the junction were flexible enough, or the DNA double strand was flexible enough, two arms on the same nanostar could bind, but the DNA stiffness prevents that from happening. And just like we had some unpaired bases in the junction here to give flexibility, we've kept an unpaired base at the bottom of each arm, up close to the arm. And that means that when the two nanostars come together, there's still some single bonds that are holding them. So there's some rotational freedom and flexibility there. When they do come together, the junctions are not very far apart. These arms are about 7-8 nanometers long themselves, and so the junctions end up twice that apart, and that distance is still very short compared to how long a double-stranded DNA would stay straight, even under thermal fluctuations. So we end up with a network of these arms, of these nanostars, and with sticky ends that are quite transient. And that allows the condensation of them to maintain a fluid character. We've been looking at that condensation, at the character of that fluid, using small amplitude oscillatory shear, because we don't make huge quantities of this fluid. The concentrations are quite high, 500 micromolar, and we can get out about 100 microliters for a few $100s. So we try to work economically with the material as we characterize it. And that constrains us to working in a parallel plate geometry in the rheometer. But this rheometry, I suppose many in the audience are already super familiar, allows us to apply strain on one plate and measure the stress on the other. And from looking at how the stress and the strain are related, we can deduce the viscoelastic moduli of the material. Now, your typical polymer solution has this type of relationship between its viscoelastic moduli and the frequency with which it's sheared. At the lowest, at the lowest end, called the terminal region, it behaves like a fluid with a scaling law in its loss modulus of just 1 over the frequency and 1 over the frequency squared in its storage modulus. But as the frequency increases, entanglements happen in a typical polymer, and one reaches what's called the rubbery plateau, where the storage modulus exceeds the loss modulus. So now it's behaving a lot more like a solid. And this crossover time is a characteristic of the longest relaxation time for polymers in that sample to rearrange, right? If you go faster still in the shear rate, you come into a transition region, which is fluid-like in the sense that the loss modulus exceeds the storage modulus. And then finally, solid-like as a glassy region happens at even higher temperatures, higher frequencies. This crossover here, which represents the longest relaxation time, is one of the interesting parameters in our materials, and it tells us something about how what we're finding is not exactly the same as a polymer solution. So this is some of this is the data for a 3-arm nanostar solution, where the storage modulus scales as expected with nearly a power law of 2, and the loss modulus as a power law of 1. And there's a relaxation time and eventually becomes fully bonded. Notice that this is happening for a fluid with a very low volume fraction of nanostars, so only 1% of the ends. Actually, this is per end, I suppose, but it's a very low, even though the concentration looks high, there's still more than 90% of this material is water. And this relationship, which shows it going from both a fluid character to a solid character, makes these coacervates into what are called Maxwell fluids. And we look at how that interaction time, that relaxation time, changes with temperature and obeys the same sort of Arrhenius behavior that others have seen in polymer solutions and all. But the interesting thing is that the activation energy encoded by this Arrhenius behavior is very similar to what would be predicted for the sticky ends energy of binding. And that, oops, get my fingers on here in the right way. That is the same activation energy regardless of valence. This tells us that although, you know, you might think of these originally from their behavior as if they were entangling, really the relaxation time is controlled by the sticky end strength. And that's the strength of a single sticky end, no matter how many bonds one nanostar is making with other nanostars, no matter what the valence. So somehow the structure of this fluid is arranged in a way that shear can relax even if one bond breaks. Now we get to the part where we play a bit of the game that I think is so fun to play once you're building things out of DNA, where we can actually change a feature of the nanostars. In this case, this is the data I showed you before for a 3-arm nanostar. And we can do the same sort of study with a 4-arm nanostar that has exactly the same length of arms and sticky end strength of arms and number of unpaired bases in the junction. We call it alpha sub 4. And maybe not too surprisingly, it ends up being a somewhat stiffer network and yet still a fluid, still a Maxwellian. And we've gone up, I keep hitting the wrong button. We've gone up to 5 arms and 6 arms under where otherwise constant conditions, but now it's quite remarkable that the fluid, the network phase, the solid character, has increased in its stiffness by an order of magnitude. That's a lot larger than you might predict, given that it has only twice as many bonds per nanostar. And this is also a feature that Maxwell predicted, but more for macroscopic networks. Wasn't at all clear if it would happen in the microscopic case. These networks are isostatic, we believe, right? Where an isostatic network is one where every junction is constrained in all of its degrees of freedom. So this arrangement of rods would be considered subisostatic. It doesn't have as many constraints as degrees of freedom, whereas just in this 2D analogy, constraining one more junction makes the whole system not deform under shear. So what's happening in our network of stars, we've been able to change the valence of the junctions to the point that all 6 degrees of freedom, all 3 degrees of motion and rotation have been frozen out. And so we're seeing the large increase in stiffness that's associated with that. Interestingly, you'll notice this crossover frequency, which was the relaxation time, it's also, well, the frequency is going down, but the time scale is going up. This is it's taking the network a lot longer to relax. And yet the network is still held together by the bond energy of a single bond. So what we understand from this is that the single bonds are still breaking And they're still the primary, it's the rupture of just one bond that is what's necessary to allow relaxation under shear. But because the network has become so much more rigid, when a bond breaks, it has a good chance of reforming again. And so it takes a lot longer for the network to find a really new configuration. So they're slower to rearrange. Now we get to an even more interesting game that we get to play in this new kind of fluid, where we can manipulate the structure further. Here's that same alpha 6 arm nanostar with all the same sticky ends and same arm lengths and junction as before. What could we do to make it have a different character as viscosity or viscoelasticity? We're going to design it to transition between the 6-arm state and the 3-arm state as a function of frequency. And we'll do this, we did this by designing an arm that had, designing a nanostar that had 2 different kinds of arms. So we kept that same alpha arm on 3 of that same alpha sticky end on 3 of the arms, and we designed a new sticky end for the other 3 arms that we're going to call gamma. We kept the proportion, the number of bonds per unit area the same, per unit volume the same, and the overall concentration of the nanostar is the same. But this material had a very different behavior. So it experiences a much more gradual transition towards the plateau modulus that characterized just the 3 bonds alone. So the green lines here represent a nanostar made up of just the new kind of sticky end. It actually has all 6 arms, but we've simply cut off the ones that were of the alpha sequence. So now those are arms that can't participate in the network formation. And this particular 3 active arm nanostar is also a Maxwell fluid with a crossover, but because of the high energy of this bond, the gamma is a much stronger bond than the alpha, its crossover is at much longer relaxation times or lower frequencies. And it's very interesting to see how the combined nanostar structure, this novel fluid, doesn't follow the alpha behavior even in the region where alpha is, even though the alphas will have not be stable, like they would have already come apart, and the gammas are the only things that are controlling the interactions, the combined nanostar network ends up being stiffer. And it maintains this stiffness with this hybrid scaling up until it becomes a 6-arm, fully constrained isostatic network with roughly the same plateau modulus, the what's it called? The not loss, but storage modulus at the high frequencies. So Nate is here and he's probably going to do a better job of describing these things, and we'll talk back and forth. But this really interesting region here teaches us what we can hope to engineer and design by creative construction of new nanostars. Did I catch it, Nate? What else would you like to add?
I think the one thing to add here is that this dynamic transition that's happening that's circled in is that it's characteristic of possessing multiple relaxation modes rather than just two. So if you can click to the next slide. Typically, if a network has one single relaxation rate, it's typically just going to be a Maxwell material. Whereas when you start introducing more and more relaxations into the network, there's more modes for the network to relax and configure. And that will thus make the storage and loss modulus start to behave in a different way. And what we indeed see is, can you click on the next slide, is if we were to take a similar nanostar to the green structure, where it's just the alpha 3s, we see that the storage modulus are indeed equal to each other, and if we were to add it, those two added moduli don't equal to the black storage moduli either. And what this is telling us is that the orange and green bonds in the black network are behaving cooperatively. So when you first form the green network, you are forming a scaffolding for the orange bonds, and as you progressively increase the frequency, more and more orange bonds are becoming bound to that scaffold, which means you're now decreasing the amount of configurational modes that the network has present because you're starting to lock the junctions. So that is one way we can start controlling the viscosity, is by essentially changing the amount of isostaticity we initially introduce into the network and the ending point of isostaticity, and that will hopefully change this interesting region where we see a cooperative kind of multi-relaxational mode of network relaxation times.
So it's because the green network is sort of holding the orange network in place, it starts to form its bonds, when if it was all by itself, it wouldn't actually form those bonds.
Correct.
So did you already say this and I didn't go forward?
Oh, yeah. So pretty much what we're trying to say here is that this kind of regime of complex kind of relaxation is only occurring between the green and orange sticky end lifetimes, and if there was kind of some independent behavior, we wouldn't be between the two bonds, we wouldn't be seeing this kind of complex kind of viscous behavior.
So by tuning the sticky ends, we can change the width of this regime.
Correct.
And maybe by changing also what else could we do to say make it go flatter? By increasing the valence, so we could end up with, instead of 3 + 3, 4 + 4?
Correct, yeah. So it's, say if you had 4 starting green sticky bonds, then that would increase the knee on the low terminal side up a little bit, right? And that would make the slope of the red line a little bit shallower. Alternatively, right, you could also make the green bond weaker, and that would then shorten the frequency regime over which the complex relaxation happens, and that would then make the slope steeper.
Cool. So this is a game that we play. Why is that not advancing? And we're hoping to explore it some more. And these different solutions are, precious, and some of the products that Rheosense has may help us to explore some of their properties with smaller quantities, letting us go through a lot more design iterations. It would be kind of fun.
Correct. Yeah, and another game that would be interesting to play is instead of putting 3 different bonds on one nanostar, actually take a 3-arm nanostar and a 4-arm nanostar and mix them together and see what would happen to the resulting viscosity. Because you can get a mixture. The phase diagram of the resulting coacervate material is going to be different, and because you're going to have different interaction strengths between the 2 nanostar's.
And you could make the experiment you're describing would have all the same, we still uniform, all the same sticky ends on everything. Mm-hmm. But if you just had a mixture of 4 arms that could interact with 4 arms and 3 arms that could interact with 3 arms, you might expect to see a phase separation as the 4 arms come together and maybe exclude the 3 arms or something like that.
Possibly, yeah.
It'd be interesting to see how it can go. So that's what we have to share with you. And since you can't see us, this right here is Nate, and that's me. And this has been done in collaboration with Omar Saleh's group. Alex Beve was an undergrad who's now in grad school at UCSD who helped out. And Chase Boders and Frank McIntosh helped with some of the theory. And Rachel is at our materials research lab helping us with using of the rheometers. Be happy to take any questions.
Thank you both so much. And as I said at the beginning, please drop your questions into the questions panel, and then Nate and Dr. Fygenson can answer them for us. And I will give everyone just 1 minute to drop some questions. And as we ask, if more come up, please again, feel free to ask. And for both of you, the first question is, what are the future applications of this technology?
Go ahead.
I think one of the first things that Deborah nailed on is that this DNA nanostar liquid is a very model system in the sense that you only need DNA nanostars to form the liquid, and the sticky ends are controlled by the sticky end sequence, right? So you know exactly how to tune the timescale of the fluid. And we also know in the sense that if we change the arm numbers, we get a stiffer network, right? So the ability to control this network gives you a wide array of parameter space to investigate what could affect viscosity. So you have a much more rational route to go down and say, oh, does arm number affect viscosity? Does bond lifetime affect viscosity? When we couple those two, how does that affect viscosity? So now we're kind of building out these little paths in a kind of darkened forest using this model DNA system. I think that's where the future applications are going to come, is where people see, okay, this is an interesting liquid behavior. Can we try and replicate this in this analog system that we're using?
I think that's very motivating, and I'm also very interested in the fact that these coacervates that one finds in biology, their functionality depends a lot on the viscosity inside of them. And here we have a system where we can create such compatible coacervates with viscosity that we might choose, and then really observe that effect on how, say, a nanostar coacervate might sequester some small molecule out of solution, and at what rate it will come together, and so how efficiently it might trap an analyte that you're interested in that's only present, say, dilute in a solution of plasma or something. You add these nanostars and you induce them to form their coacervate, and they perform a kind of concentration of dilute analyte that you might not be able to realize in another way. So that's another common line of reasoning and application that people have proposed for the nanostar system.
And someone's asked, how can torque overload be prevented in the rheometers?
I typically don't go to high enough frequencies. I think that's when the geometry inertia comes into play. And also, if your material's too stiff, that could then ruin the measurement you're doing. We are in a regime where we're actually pretty low viscosity for bulk rheometers. So we have to use a pretty large flat plate compared to most materials. But I think that's the answer.
So torque overload isn't something that has ever happened.
Yeah, I haven't noticed that at all in any of our materials. They're not too stiff to affect the rheometer's kind of ability to torque, and then also I don't go to high enough frequencies such that the rheometer starts shaking around crazy.
And have you ever looked at phase angle delta versus frequency? You may be able to identify the different transition regions.
Yes, so I have looked at that like with the very cursory eye on the complex nanostar structures that we looked at. You can definitely tell that there is a specific phase angle for each of the regimes, right? So you have obviously pi halves for the fluid regime, and then once you get into the complex kind of scaling regime, the delta definitely stays at a constant value through that regime. And I believe I actually did use that to fit the power law. So the answer is yes, I have used that in my analysis.
And are you planning to study concentration variance or ratio between stars?
So we have studied concentration variance in our first paper. So we've taken the 3-arm nanostar and varied its concentration and measured the power law as a function of concentration. The stiffness, the network stiffness as a function of concentration. And we did that for 3, 4, and 5, and 6.
I wonder if that's in the supplemental that on the slides.
But basically what we see is excuse me, with the 3-arm nanostars, we see a concentration power law with stiffness that's pretty consistent with a floppy percolated network. And then once we go to a 6-arm network, we see a network stiffness scaling with concentration that's consistent with just afinely deforming rigid strands. I think Deborah's going to throw that plot up there right now. Yeah, there we go. And interestingly, you'll see that there's not a clear monotonic kind of change in that concentration power law. It goes from 1.8 to 1.3 to 1.5 to 1.0. And that's kind of denoting that the 5-arm nanostar doesn't really have a defined geometric ground state. So it's probably frustrated a bit trying to find a ground state, and it has a little bit more configurational freedom than the 4-arm nanostar in terms of how it scales with concentration.
Someone's wondering, could these nanostars be considered building blocks for molecular level structures, DNA-based machines?
Yeah, I think so. I think Deborah can answer that a little bit better.
The nanostar's structure is fairly simple and floppy. And so to make a machine, you want to have ability to cycle through different structural states under control of some energy input or release. So the nanostars, there's lots of ways to fold up DNA and make more complex shapes that are more likely to form the part of machinery. The advantage of the nanostars, because of their simple design, is that we can make them in relatively large quantity compared to the more elaborate shapes. And so we can start to interrogate real material properties. I see the nanostar sort of coacervate as the technology here, not something at the single nanostar level.
Yeah, so you could think about maybe computing coacervates or making a kind of computation with coacervate structures, right? Say the coacervate A, what does it do with coacervate B? Can they mix together or can they not mix together?
Oh yeah,
and you can start
engulf one another.
Yeah, and then you can start building off logic from that possibly. But I think
but we don't know yet how to inject the energy into them. Right. So that'll be an interesting.
There's a lot of work in trying to transcribe DNA out of nanostar coacervates right now, and that's what some of Omar's graduate students are trying to do. So there is some progress in terms of trying to get out get
genetic control going.
Genetic control over the nanostars. So if you consider that as computation.
Well, the challenge there is really that this environment, this viscous fluid, is doing stuff to the reaction rates that we're more familiar with in just sort of cytoplasm or more aqueous environments. So it may be very challenging for the enzymes that we want to have work inside of the nanostar coacervate as it's currently constructed. But we have so much control, we could go to longer arm lengths and therefore make the pore size in whatever network is forming a lot larger. Or we could introduce more flexibility between the arms. So there's going to be a back and forth between enabling sort of larger scale changes in the fluid from the control, and just how much the fluid manipulates the controlling particles. These are nice questions. Thank you.
And someone's wondering, do you or have you performed rheology of crude oils and gas condensates in your group as well?
No, we have no experience. We're quite the dilettantes when it comes to rheology, and this we're sort of learning as we go. This is our first work in rheometry. We started wanting to do more micro-rheology and found that the structures were just too viscous and not amenable to that. We do a lot more work microscopically. So we had to go to the full-on rheometer.
And I'll give everyone just 1 or 2 more minutes to see if any additional questions come trickling in. So as we wrap up, just a reminder, if you would like to connect with our presenters for the day, you can always reach out to our team and we're happy to help connect you. And I just want to say thank you again to Dr. Fygenson and to Nate. Oh, of course, as I do this without fail, every presentation we get another question. Have you applied Mills's law of monodispersive suspensions in your DNA work?
No, Mill's law is not something I'm familiar with. Our suspensions are not monodisperse. The nucleation rate of the coacervation is a very interesting problem, and how one can make it happen. Right now, all the data we showed you was in already a condensed phase that had already sedimented sort of out of solution, so that we had the very high concentrations needed to be uniform. Not really sure. Can look into what that is. If you wouldn't mind contacting us, happy to learn more.
The response to that was, it was originally developed for red blood cells, but it's been extended to emulsions.
Yeah, the fluid we're making is entirely a dense coacervate phase. So in the rheometer, we throw in 100 microliters of the coacervate phase. So we essentially just make a very dense fluid from the get-go. Other ways you can make the fluid is by extracting out the supernatant from the phase-separated solution, but we went ahead and just made the dense coacervate phase by just throwing a bunch of DNA in the first place, because that throws us outside of the immiscibility region of the phase coexistence region.
Right. So the questioner is probably thinking that one is in this region of droplet coarsening or ripening, when we're really down here in the fully phase separated region and taking just this coacervate phase into our studies.
Wonderful. Well, again, I want to thank Dr. Fygenson and Nate Conrad for joining us today. Thank you so much for such a wonderful presentation. And as we move into our last speaker of the day, we're going to take a 15-minute break, and then we will conclude today's event with our 2 p.m. presentation from Dr. Stacey Elliott with Rheosense. So thank you again, Dr. Fygenson and Nate.